One obvious difference between the human genome and the genomes of other great apes is the organization in chromosomes. It has been known for several decades that the karyotype, the arrangement of chromosomes according to their size and morphology, is different in humans and in other great apes. The most striking difference, as you see in this ideogram showing all the chromosomes in 4 great apes, human, chimp, gorilla and orangutan, is that human chromosome 2 is the result of a fusion of what in the other 3 great apes are 2 separate chromosomes. In addition to this gross rearrangement, there are about 1500 inversions, many of which are cryptic. That means that they are not visible at the microscope because they are very small. And in total, it is estimated that up to 90 megabases of sequence are included in various inversions and deletions. Of course, they might be just irrelevant in evolutionary terms, or they could change the expression of genes, their activity, in ways that might have profound implications for the evolution of our species. It is not yet known, but some recent observations have shed some light on this. You will remember that the duplication of genomic segments is one of the main mechanisms of genome evolution. Well, previous studies about the evolution of some gene families indicated that the gain and loss of genes were particularly frequent during the evolution of primates. Comparing the genomes of humans with African and non-African great apes, scientists discovered that there was a sort of burst of duplications in the genomes of humans and African great apes. And interestingly, these duplications were enriched in genes involved in brain developmental processes. Recently, one of these duplications was studied in more detail, and the results are striking. There is a gene known as SRGAP2 that is found in 4 different copies in the human genome. We acquired these 4 copies by 2 duplications from one original copy that is called SRGAP2A. What happened was that a first duplication created version B of this gene, SRGAP2B, and that's the name of this new copy. Later, this B form duplicated twice independently to originate forms C and D. And what is interesting about this family of genes is that they control the formation of synapses, the connections between neurons in the brain. The normal function of the original copy, SRGAP2A, is to prevent synapse formation in the cortex of the brain. And in fact, some problems with this gene had already been reported in a girl with a type of intellectual disability. But then, the first duplication interrupted the sequence of the gene so that SRGAP2B, the new copy, lost its function, but one of the other copies, SRGAP2C, seems to have acquired a new function with exactly the opposite effect to the original copy, the A copy. We know this because scientists introduced SRGAP2C in mice. Mice only have the A version, SRGAP2A, because these duplications, remember, took place in the human lineage. So what happened in the brain of mice after the introduction of SRGAP2C in their genome? Well, surprisingly, synapse formation accelerated. It counterbalanced the effect of the normal SRGAP2A gene. Increasing the number of connections in the brain is, in a sense, a bit like increasing computing power. So this genetic innovation might have been very important during the evolution of the human brain. So the question is, are they really related to human evolution, these genes, these changes? Well, it depends on when these duplications took place. It's difficult to know. But the good news is that comparing the sequences of the 4 human copies, scientists could determine the age of each copy quite accurately. And you know what? Well, apparently, the first duplication, the one that created SRGAP2B, occurred around 3.4 million years ago. And the second duplication, the most important one because it originated SRGAP2C, the copy that increases computing power in the brain, occurred around 2.4 million years ago. And this is very close to the age of the first recognizable stone tools made by humans about 2.5 million years ago. And also coincides with the increase in brain size that we see around that time in hominin fossils. So when our brains got bigger, synapse formation would have exploded thanks to this duplication. Intriguing, is it not?